can cause bloody diarrhea. One you have already read, Escherichia coli. There are others as well, but today we are going to talk about the commonest cause of bacillary dysentery, that is shigella. Okay, so actually, shigellosis has been described from biblical times. So it was known as the bloody flux. Okay, so shigellosis is also known as the bloody flux. Bloody flux. Why? Flux is discharge, right? So the stool that comes out is mixed with blood and mucus. So it has been described as the bloody flux of biblical times. So the agent or the bacteria, it is it belongs to the family Enterobacteriaceae. There are other bacteria, other tribes and genuses belonging to Enterobacteriaceae. Some of them you've already read about, like Escherichia coli. You'll also be reading about Salmonella, and today we'll be talking in detail about the genus Shigella. Okay, so Shigella is a gram-negative. Enterobacteriaceae actually is a gram-negative, is a family of gram-negative bacilli. So Shigella is also a gram-negative bacilli. Most of the Enterobacteriaceae are motile with peritrichous flagellae, but Shigella is a non-motile bacteria. It does not move. So in the TSI, the triple sugar iron medium, or if you do a hanging drop, you will not see any movement. Okay, and this is non-capsulated. Important here is these are non-lactose fermenting. So when a patient comes with complaints of dysentery, and if you proceed in the lab to isolate the bacteria, you use McConkey agar. McConkey agar is a differential agar, right? Differential medium, where it differentiates between lactose fermenting and non-lactose fermenting organisms. So if you find in the McConkey agar transparent, colorless colonies, then those are known as the non-lactose fermenting. The lactose fermenting ones will appear as pink, right? So amongst the bacteria that you'll be reading in your GI system, Shigella is a non-lactose fermenting. Salmonella also is non-lactose fermenting. Whereas Escherichia coli that you read is a lactose fermenter. So the colonies of Shigella will appear colorless. Also in TSI, there will be fermentation of glucose, but there will be no gas formation. Similarly, the bacteria, they cannot decarboxylate lysine, nor can they hydrolyze arginine. Lysine, arginine, and ornithine, these are three amino acids. Again, amongst the battery of biochemical tests that you can put up for identification of bacteria, these three are the amino acids. So few of the bacteria, they are able to decarboxylate lysine and ornithine. Some of them, they will hydrolyze arginine, while some of the others will not. So depending on the biochemical reactions, you can identify the organism. Okay, so in case of Shigella, all the Shigella, they will not be decarboxylating the lysine, and they will not hydrolyze arginine. Whereas one of the Shigella group will cause decarboxylation of ornithine. So we come to the groups. The picture here is of Shiga, the scientist, Kiyoshi Shiga, who in 1898 first discovered the agent that caused bloody diarrhea or dysentery, and the organism that he discovered was later named from him, and it's now known as Shigella dysenteriae. So there are amongst the 40 different serotypes that can cause infection in humans. Remember, the Shigella, they cause infection in the primates and humans. Okay, they do not cause infection to others. So they are basically reservoirs in the humans and in the other primates. And of the 40 different serotypes, they can be divided according to the antigenic property and according to the biochemical reactions they produce into 4 different groups. In group A, we have the agent, the species of shigella that was first discovered in 1898 by Kiyoshi Shiga, and that we call it as Shigella dysenteriae. And it has 1 to 12 serotypes. The second group, group B, is it contains of Shigella flexneri species. Again, the name flexneri comes from the person who discovered it first in 1900, Flexner. Shigella flexneri has 6 different serotypes, 1 to 5. It has other variants like X and Y variants, and other varieties like variety Newcastle, and variety Manchester. Then we have the third group, which is discovered by Boyd in India, and the name given to the species is Shigella boydii. There are 1 to 18 serotypes, so 18 serotypes. And the last is Shigella sonii, which was identified and discovered by Sonii in Denmark. And here it has a single serotype. Serotyping is the antigenic diversity, right? It has a single serotype, but the collicin that it produces, they are different. So depending on the collicin production, we have different collicin types in case of Shigella sonii. And there are 1 to 17 different collicin types in Shigella sonii. So please remember these 4 groups and their names. The first one is Shigella dysenteriae, which causes the most severe form of dysentery. Then we have the second group, which is Shigella flexneri. Shigella flexneri is the common species that is found in Nepal. Then we have group C, which is Shigella boydii, and group D, which is Shigella sonii. Now, this Shigella sonii, it is an exception in the sense we said that Shigella, they are non-lactose fermenters, right? But Shigella sonii, it can cause a late lactose fermentation. So if you keep the colonies of Shigella sonii that has grown on the McConkey agar plate for a few days, it will turn into pink. That is because they will ferment the lactose late. So Shigella sonii is late lactose fermenter, and it has collicin types instead of serotypes. So this is to stress on the different biochemical reactions which are unique to each of these groups. The first group, Shigella dysenteriae, these are mannitol non-fermenters, whereas the rest of the other 3 groups, they can ferment mannitol. So if you put up the reaction mannitol fermentation test, then the Shigella dysenteriae will not ferment mannitol, whereas Shigella flexneri, sonii, and boydii, they will ferment. Then out amongst the 12 different serotypes of Shigella dysenteriae type 1, it is catalase negative. It does not cause the formation of bubbles when you add a drop of hydrogen peroxide over the colony. So these are catalase negative Shigella dysenteriae type 1. Then we said in the general properties that they ferment glucose, but they do not produce gas. Again, this statement has some exceptions. So the species that produce gas from glucose are Shigella flexneri, the 6th variety, and variety Newcastle, and variety Manchester. We discuss about Shigella sonii being the late lactose fermenter, and also Shigella sonii, it can decarboxylate ornithine. So these are the different exceptions from the broad properties of the genus Shigella. Of this, what you should remember is Shigella dysenteriae type 1, which is catalase negative, mannitol non-fermenter. And these are the ones which cause severe kind of dysentery, which can also lead to death. So a little bit more in detail about this Shigella dysentery type 1, because rest of the Shigella infections, they are self-limiting. In adults, they are self-limiting. So without antibiotics, without other treatment also, they contain themselves after a few days. Whereas Shigella dysentery type 1, they are associated with certain complications. So we'll be dealing with dysentery type 1 a little more in detail. So Shigella dysentery type 1, it has been associated with large and prolonged epidemics of, I should say, dysentery. Okay, and another important thing about Shigella is that all the Shigella, not just dysentery, it's more it is very capable of picking up antimicrobial resistant plasmids. So antimicrobial resistance is quick and more frequent to develop. Shigella dysentery type 1 causes more severe, prolonged, and fatal illness. So how do we, I said that this is dysentery, right? Dysentery is diarrhea with blood and mucus. In Nepali we call it आउँपर्ने. आउँपर्ने भनेको चाहिँ पेट दुख्छ, घरिघरि टोयलेट गइरहनु पर्ने. So dust wala tatti for India, Indian. Dust, no, bloody diarrhea, what do you call it? Okay, whatever. Then, so how does it, what is the pathogenesis? I've told you that they can only infect the humans and the primates. The infective dose is very low. Okay. So if we see in case of cholera and in case of Salmonella typhi causing typhoid fever, the infective dose, the number of bacteria required to cause an infection, is quite high, around 10 to the power 6 to 20 to the power 8 in those cases. Whereas in case of Shigella, it even 10 to 100 organisms, they are capable of causing infection. So that's why these Shigella, they can be easily transmitted from the 5 Fs, the fingers, fomites, flies, food, and what's the next? Whatever. So very less number of organisms are capable of causing infection. That's why there is a large number of secondary cases if someone in a household gets infected. And the incubation period is short, around 2 to 3 days. The clinical features, it will be a sudden onset of abdominal colic, pain, cramps, tenesmus. There is rectal spasm when you go for defecation. And the stool has a small volume. Initially, it will be watery diarrhea. Then later on, it will be just mucus and blood mixed stool. And there will be associated fever as well. Whereas in case of the other type of dysentery that I talked about, in case of the amoebic dysentery, you will not have fever. The number of stool passes is also less. Okay. And it can last around 4 days to 1 weeks. The bacteria, the Shigella, they are ingested. I told you the mode of transmission is by either finger, flies, fomites, food, right? So it is ingested and they are not destroyed by the gastric acid or the bile, and they reach the large intestine. In the large intestine, the receptors on the intestinal lining, the glycoside, they invade the large intestine by inducing the endocytosis, and within the intestinal cells they will multiply and they uh, invade the adjacent cells. They cause inflammation in the lamina propria. There will be capillary thrombosis. There will be release of some toxins, exotoxins known as the Shiga toxin. They cause the protein synthesis inhibition, which leads to necrosis, and then there will be superficial ulcers. So besides the superficial necrosis and superficial ulcers, exotoxins are also responsible for the water and electrolyte secretion and causation of diarrhea and blood mixed diarrhea, dysentery. So this is what is happening. The bacteria, they reach the large intestine, and through the intestinal cells, mainly the M cells in the intestinal lining, They will induce endocytosis. So these are engulfed into the M cells. Then they will come out of this endosome and they will multiply. They use the actin filaments within the cell to enter the adjacent cells. So thus in the adjacent cells are invaded, they will multiply there again, and then they will come down. Here they are either engulfed by macrophages and other lymphocytes, and even the cells which are infected, they get cell lysis and inflammation. This picture will show you in more detail how they enter. So there is this bacteria, they enter via the induced endocytosis. The, the cells will multiply within this infected cell, and they will cause actin polymerization, and they will move from one cell to the adjacent cells. So they will cause there is intercellular dissemination of the bacteria. Shigella dysentery produces a type of exotoxin as well, which is known as Shiga toxin. You must have read in E. coli some Shiga-like toxin, but they have the similar kind of mechanism of action. So Shiga toxin, also known as the virotoxin, they act in they inhibit the protein synthesis. That's why there is necrosis and superficial ulcers. So the toxin, you must have read in your class on respiratory system about diphtheria toxin, right? Diphtheria toxin also causes protein synthesis inhibition. But diphtheria toxin it is encoded by the lysosomal phase, whereas this toxin, Shiga toxin of Shigella, it is encoded in the chromosome itself. It's not encoded by the bacteriophage or the plasmid. It's chromosomally there. The structure is it has 1 unit subunit A and 5 units of subunit B. So it's a toxin which has A and B subunits. The B subunit, it is for binding to the host receptor, and the A subunit, it is the active component which causes the damage. And the A1 subunit, it will inactivate the 28S ribosomal RNA to inhibit protein synthesis. That's why you'll see abscess formation in the intestinal lining in the large intestine, and there will be necrosis. Later on, there'll be superficial ulcers, and then in the damaged mucosa, you can see some pseudomembrane also lining it. After the ulcers heal, there will be scarring. And when the toxins act on these small blood vessels, there will be microangiopathy, and that will result into a complication known as HUS, or hemolytic uremic syndrome. So hemolytic uremic syndrome, it causes the microangiopathic hemolytic anemia. It causes thrombocytopenia, platelets are reduced, and it causes acute renal failure. So all these three, the triad, is known as HUS, the syndrome, hemolytic uremic syndrome. Also, Shigella has been associated with extra-intestinal complications. The Shiga toxin, it has neurological effects as well, so it can cause neurological disorders. Reiter syndrome, it's known as the reactive arthritis, where you'll have mainly in people with HLA-B27 uh, uh, chromosomes, then they can cause Reiter syndrome, which is conjunctivitis, urethritis, and arthritis. Seizures have also been reported. The bacteria is very rarely found in the blood, but sepsis has again occurred because it is the defecation is very painful and there is tenesmus, and one feels like going and to the loo and causing like evacuating, that there is chances of rectal prolapse. And toxic megacolon has also been associated as one of the complications. So how do we diagnose? I told you we have to differentiate from amoebic dysentery. Few of the history, how it progresses, whether there is an associated fever or not, what was the stool quantity, the frequency, right? Is, all these also help in the differentiation. But for the definitive diagnosis, if you want to isolate the bacteria and know the species, what is done is you have to culture the bacteria and identify it. So, what are the samples that are commonly taken? The specimen is stool, and when you collect the stool, if you take the mucosal, the flakes of mucosa that is shed, then that is better. Rectal swabs can also be taken. It has to be transported immediately to the lab. If there is a delay, then it should be transported in transport media. Usually, the transport media that is used is the buffered glycerol saline, or you can also use Gary Blair, Amy's, or Schwartz medium. Then you can enrich the stool using some enrichment medium known like the gram-negative broth, GN broth, or the selenite F broth. So the enrichment medium, it suppresses the other normal flora of the stool and allows the multiplication of the shigella only. Okay, so within, if you put a loopful of the specimen in this broth, selenite F broth or GN broth, after around 4 to 6 hours, you have to subculture the broth onto the solid medium. After 6 to 8 hours, rest of the flora from in the stool, they can also start multiplying. So you have to do the subculturing after around 4 to 6 hours. So the stool specimen, they should be promptly plated. The plating media that are used are the selective media like McConkey and one of the two other selective mediums like the XLT, which is xylose lysine deoxycholate medium, and the DCA, deoxycholate citrate agar. So these selective medium, they will inhibit the growth of gram-positive bacteria and other enterobacteriaceae. It will allow the growth of Shigella. Then after you find non-lactose fermenting colonies in your plate, then what you should do is you should perform the biochemical tests, like you could inoculate the triple sugar iron, TSI, and the motility agar. Then in that, if you see there is no gas production, there is acid like fermentation of sugar glucose producing acid, but no gas formation, and the organisms are non-motile, then you proceed to serological identification. So how do you perform the serological identification is we have the polyvalent group sera. So the grouping sera is antibodies against the O antigens. Okay, so the O antigens are the somatic antigens, which are lipopolysaccharides. So the O antigens, the antisera, which are commercially prepared against the polyvalent O antigen, is used to find out first whether they belong to the genus Shigella or not. So if there is an agglutination, if the colony that you have grown, it agglutinates with the uh, polyvalent O antigen sera, then we confirm that it is Shigella. Then we have to group them into 4 different groups, whether it is Shigella dysenteriae or Shigella flexneri or Boydii or Soniae. Your biochemical reactions will help in uh, grouping them. Then accordingly, we'll again use the sera to check for the agglutination. So this is how we proceed. If we find colorless, non-lactose fermenting, non-motile organisms, and there is only acid, there is no gas formation, we put up the mannitol test. If it is mannitol negative, mannitol is not fermented, then we call it as Shigella dysenteriae. So to confirm it by serotyping, we can use the group specific, the first group, group 1 specific antisera. And then if it is mannitol positive, we look for the fermentation of lactose. If it is a late lactose positive, then we group it under the Shigella sonnei. If they are lactose negative, we look for the indole reaction. If it is indole negative, it is Shigella boydii. If it is indole positive, it is Shigella flexneri. So this is how you, and then again, for each of these, you have to confirm it. This is just phenotypic identification, right? Phenotypic by the properties of the biochemical reactions. So then you do the serotyping. So you take the monoclonal antibodies of each group and then do the agglutination. If they agglutinate with that specific antisera, then we call it as belonging to that particular group. So how do we treat the case? It is usually self-limiting, but the severe form, which is caused by the Shigella dysentery type 1, it requires treatment. The WHO recommended drugs are usually ampicillin, cotrimoxazole, naloxone acid. We keep the ciproflox and norfloxacin as reserve drugs. And as we've said that the Shigella, they can acquire the resistance, horizontal transfer of resistance through the plasmids. So plasmid-mediated multi-drug resistance is quite common in Shigella. Also, chromosomal resistance to quinolones have been reported. So you have to be careful while treating the cases. There is no efficacy in vivo of aminoglycosides, amoxicillins, first and second generation cephalosporins, and nitrofuran joints against Shigella. So this is a frequently asked question, and it is important in clinical diagnosis as well. The differences between amoebic and bacillary dysentery. Bacillary here means the Shigella dysentery caused by Shigella. Incubation period is short in case of bacillary dysentery, whereas it is longer in amoebic dysentery. Fever is absent in amoebic dysentery, while bacillary dysentery, the fever is there. The volume of stool that is passed is small in case of bacillary dysentery, whereas in case of amoebic dysentery, it is copious. Actually, in bacillary dysentery, in the initial stages, when the bacteria has not invaded yet, it is only the toxin that is causing the loose motion. Then it is more of watery diarrhea. Then after 2 or 3 days, then it will start to have blood and mucus, and there will be very little volume of stool that is passed. Okay. Then the frequency is also more in case of bacillary dysentery, but again, these are all subjective. The thing that will clinch the diagnosis is the microscopy. In the microscopy, you will see that the pus cells in the stool are few in case of amoebic dysentery, whereas there will be plenty of pus cells and few RBCs in bacillary dysentery. In case of amoebic dysentery, you'll also be able to see the entamoeba histolytica cysts or trophozoites if it is a very fresh stool. Also, the normal flora of the stool will be intact in case of amoebic dysentery, whereas in case of bacillary dysentery, there will be reduced normal flora. So these are how you will be able to differentiate between the amoebic dysentery and bacillary dysentery. So to avoid getting infected, this is the rule. Don't eat anything raw. Boil it, cook it, peel it. If you cannot do any of these three, forget it. So that's how you avoid getting Shigella dysentery. And the practice of hand hygiene is always good to follow. Okay, so to summarize, we've seen that the Shigella dysentery, the dysentery caused by Shigella, it can be caused by any of the four groups Group A, which is Shigella dysenteriae. Group B is Shigella flexneri. The third group is Shigella boydii, and the fourth is Sonii. We have different biochemical reactions common to all the Shigella, like they are non-fermenters, and they will ferment glucose without the production of gas, and they are non-motile, and they usually do not decarboxylase lysine or hydrolyze arginine, but there are